to my curious little children. I hope I am audible as well as visible, but let me just check. Dun-dun-dun. Yes. Oh my god. Okay. So as and when you come, we will start off this particular session. Let's just wait for some time. By the way, everyone who probably might be watching it towards the end, as in after the live ends, I hope that you guys are doing good. I hope most of your classes have already been started. Well, if you face any trouble in this particular chapter, do let me know. I could be helping you well. Okay? Cool. So while children come, let's start off this particular session. So before this, I have done 3 videos, 3 live lectures on the same chapter, that is sexual reproduction in flowering plants. The first part was about the male reproductive system. I spoke about everything, the microsporogenesis, the formation of male gametophyte, that is your pollen grain, everything. In the second session, I spoke about megasporogenesis, as in we spoke about the female reproductive part, how megaspores are being produced, how embryo sac is being formed and everything. And in that particular video, I spoke about this thing, that is double fertilization. Spoke about is being repeated, I know. But yes, double fertilization was dealt in the second session. Now the third session we we discussed basically the pollination, the methods of pollination. Apart from that, we also mentioned the artificial hybridization. We also mentioned one more particular important thing, that was what artificial hybridization and outbreeding devices. So these were the few things that we already covered. In the last part, that is today, we are gonna cover endosperm formation, embryo formation, and we will see the different types of seeds which are being produced. We'll talk about different techniques, or basically different processes which are happening in the plants. For example, apomixis, polyembryony, parthenocarpy, which are something that usually confuses children. So this is what I have for you, and with that, let's go ahead with today's session. So we'll just have a quick recap of what exactly is double fertilization, otherwise you will not understand anything. So listen to me very carefully. This particular thing that you see right over here is your embryo sac. What is it? It is the embryo sac. And where is the embryo sac? So ma'am, I know that over here you have the ovary, inside that we have the ovule, inside the ovule we have what? The embryo sac, where the female gamete is there. Okay, so this is the embryo sac, this is the ovule, and this is the ovary out there. Whenever we say that fertilization has taken place, after fertilization, ovary is not known as ovary anymore. Ma'am, then what? It is known as a fruit. Okay, what about the ovule which is there? Ovule is known as the seed. And what happens inside the embryo sac? The embryo is going to be formed. That is why we say that the seed has the embryo. That is the basic logic. So as of now, I am deep inside, and over here, this is the structure of the embryo sac. In the embryo sac, there are unique features about it, that there are 7 cells, but there are 8 nucleus in them. There are 7 cells, but there are 8 nucleus. And if at all you have confusion, then listen to me out carefully. This is 1 cell. Okay. The next cell. How many cells are there? 3 cells, done. And 3 of them have their own nuclei. The green ones are basically nothing but their nuclei. What do I call the cell? Ma'am, I call them as antipodal cell. All cool. What happens to them? There is no much information about them, so you do not need to be worried about them also. What's more important is right over here. You see these 3 cells? They are best friends. These 2 cells over here, which are in purple color or pink color, they are known as what? Synergid cell. What do you call them? Synergids. And over here you have the main lady, and that is what? The egg cell, which you also call as the female gamete. So this is what? Your female gamete. Since female gamete, that is the egg cell, is along with synergids, basically we say that all of them are best friends, so we say that they are egg apparatus, or it's a part of what? The egg apparatus, which you see right over here. Cool. Yes. Now what about these 2 things? These are nothing but the nuclei. Nuclei of what, ma'am? So 3 plus 3, 6 cells are already done. You told that, ma'am, you told that there are 7 cells. Where is the 7th cell? So this whole thing that you see over here, this is a part of a very big cell, which you call as what? The central cell. And the central cell, which is very big, it has 2 nuclei. And what do you call this nuclei as? two polar nuclei. They are what? Polar nuclei. So 7 cells are there, but since we have an extra nucleus, we say that it is 8 nucleate in condition. 7 celled and 8 nucleate in condition. What are we talking about? We are talking about the embryo sac. Okay, ma'am. Now, one more very important thing that I want to discuss over here and right here is that all the cells that you see are haploid. That means if I say normal cell in a plant is diploid in nature, or if I say that they have 48 number of chromosome, then I'll say these cells that you see over here are haploid, half of it, and they will be having only half the number of chromosomes. Cool. So this is haploid, this is haploid, this is haploid. What about them? They are also haploid. All of them are haploid except the central cell. Central cell is not haploid. Can you guess why? Because over here the nucleus has half the number of chromosome. Here also the nucleus has half number of chromosome, but they have 2 nuclei, right? So n plus n, they already become what? 2n. So central cell which is there, it is diploid in nature, just like a normal cell of the plant. It is the only one which is diploid in nature. Now the reason why I spoke about all of this is right now fertilization is gonna happen, and I'll be talking about that, and then you will get the whole picture to be very, very clear. So what exactly happens is that a pollen grain which is there, it comes and lands on the stigma of the plant. Now while it does, you need to understand the pollen grain contains gametes. How many? There are 2 gametes which are there inside the pollen grain. One gamete says that, hey, my lady is right up there, I just want to go and fuse with that. So my lady is what? The egg cell. So egg cell which is there and one male gamete which is there, both of them fuse together, and that is how you get the zygote. You get what? The zygote. Your egg cell was having haploid number of chromosome. The male gamete which is there, it also has haploid number of chromosome. N and N comes together to form what? N and N, ma'am, everyone knows, even though we are not that great in mathematics, we know that it is 2N, and that is the ploidy of what? Your zygote. Zygote is a single cell. This zygote later divides to form what? It divides to form an embryo. So ma'am, first fertilization is done over here. Yes dear, it's done over here. What do you call this fusion as? You call this fusion as syngamy. You call this as syngamy. Now ma'am, you said that one more is there, right? What happens to that? The one more which is there, the one more which is there fuses with the 2 polar nuclei right over here. So another male gamete right over there fuses with the central cell which already had 2 polar nuclei. Both of them are in haploid number of chromosome, that is, it is already diploid in nature. Here this guy is haploid in nature. 2n plus n, ma'am, we know it is 3n. That means what? Triploid. It's not diploid, it's not haploid, it's triploid out there. And what do you call this thing that is being formed over here? Primary endosperm cell. You do not call it endosperm, even though I have written it like that. You call this as what? Primary endosperm cell. This is just a cell which is being formed, and the nucleus of this particular cell is known as primary endosperm nucleus. So primary endosperm nucleus, you can also call it as PEN, and basically it is triploid in nature. Now 2 fertilizations are done. This is a fertilization, this is also fertilization. You call this a syngamy. What do you call this? You call this one as triple fusion. Why triple fusion? Because 2 nuclei over here, 1 nuclei over here, 3 nuclei are fusing together. So that's why you call it as triple fusion. Right? Got it? Now, the question is, well, why did plant do all this drama? Zygote is more than enough now. Why there was a need for endosperm? The reason for that is, I told you zygote is a single cell. It has to form an embryo. The embryo forms a new plant. That is how it goes on. Now, who is there to provide nourishment to this papa guy embryo out there? It is the primary endosperm cell. The primary endosperm cell, each cell which is there, it is going to divide. They're going to form a tissue which is known as endosperm. And this is something that is going to provide nourishment to the zygote and say, Baba, I am going to take care of you. You are going to form an embryo one day. So always remember, this is being formed. But which one is the one that is going to develop faster, or the first? It is going to be the primary endosperm cell. The endosperm cell divides to form a tissue, and that is known as the endosperm. Hello Anu, Harmat, and hello Prajwal. I hope you guys are doing good. Got it? Everyone who is watching this video, which I'm pretty sure it's not too many, because I know you guys are not very much active right now. You will be active more towards examination, and that's okay, but you will understand and you will come back. My dear children, when you come back, you should be having content to study, right? So that's what I'm doing over here. Hello. Suvarna, hi. Vivek, hi. So cute of all of you. Okay. Okay. Hello, Vivek. Hello. Oh my God. Hello, Sai, sorry. Okay, anyways, so I hope you are understanding this. If at all there was someone who heard me before, what all things I was saying, can you just give me a thumbs up if at all everything is clear up to what I've taught now? Myself, Vaishu ma'am. Hello, Vaishu. So I'll call you Vaishu. I'll try to remember that name. Vaishu. Cool. If it's clear, can you give me a thumbs up? Or shall I just go forward with endosperm, because I think all of you are new and you might have to listen to what I have told before. Okay, Vaishu gave me a thumbs up, so I'll go forward. Okay, so we are going to see endosperm, and we'll see why does the endosperm form first. Because I told you the papa zygote which is there, it has to form the embryo. It has to become multicellular, and for that it needs nourishment. Who provides that? Endosperm provides that. So always listen to me. There was one nucleus which was known as a triploid nucleus, that is this known as primary endosperm nucleus. This nucleus is going to divide rapidly. Okay, so many nucleus are going to be formed over here. When I say that these nucleus are formed, you need to understand one very important thing. The nucleus is not covered by a cell wall immediately. Some of them, they just stay like that. Some of them basically are covered by cell wall. So some machas are like, I'm cool, I'll be just there. Okay, some are totally covered by what? The cell wall are there. And what do you call them? Do you have a different name for all of them? Yes, listen to me. The first one which is there, free nuclear endosperm, that is known as this thing that I have just made over here. This free nuclear endosperm is something that is not covered by the endosperm. Then we say that the cellularization has taken place, as in they are covered by cell wall and things like that, and that is how you get this cellular form of endosperm. So all of you, I'm pretty sure you might have had coconut water in your life, or if you have seen coconut, I don't think none of you have not seen coconut. Everyone might have seen it. So coconut, just imagine a coconut is there, macha, I have broken it up, not looking like this. There is a white thing, okay, there is a white thing which is there, and there is water, right? That is how it goes. So I'm not a Picasso, but still I'll draw and show it to you. So there is white thing which you call as what? You call this as kernel of the coconut, and there is water which you actually drink. Do you know what does this water contain? This actually, the water that is out there is nothing but the nucleus which is not covered by any cell. Rather, over here also you have nucleus, but what happens to this nucleus? This nucleus is being covered by cells out there, and these cells make the white kernel that you yum. That means that you eat. You get it? So this is very, very, very important. Okay? Cool, ma'am. I came and all have come. Oh my God, Anu might I'm pretty sure you have so many fans. Okay, free nuclear endosperm, exactly, Vaishu. So that is what you call it. Always remember, you had a primary endosperm nucleus. So there's two nucleus out there of the primary endosperm cell. What happened to this nucleus? This nucleus kept on dividing, but it was not covered by cell wall. So we call this as free nuclear endosperm that is being formed, and you find in coconut water. Okay, now there are certain nucleus which is covered by cell wall and which you call as cellularized form of endosperm, which is something that you eat sometimes. Cool? Yes. Now that is a basic thing that we have. Sometimes you need to understand that the fate of the endosperm is different. That means what is going to happen with endosperm is two different scenarios. Either they can be used up. Used up by whom? So as I was saying, there is a zygote. Zygote is forming an embryo, and basically endosperm is providing nourishment for that. Sometimes what happens is that the endosperm is completely used up by this embryo while it grows up. Okay, and there is another time where sometimes it says that macha, I'll have just little bit of endosperm, little bit, let it be there. So there are two ways. Sometimes it can be completely consumed before the seeds becomes mature. For example, pea, groundnut, beans, mostly all this thing that you see where they are mostly consumed completely is in the case of dicots. I am saying it is mostly, I'm not saying all the dicots. What do you mean by dicots? my baba dear children, just imagine, okay, over here you have peanut, okay, or groundnut which is there. You can break it down into two, right? You can break it into two. So you usually call them as what? Dicots. And dicots are usually the ones where the endosperm is completely utilized for the formation of the embryo. In other case, mostly there are, okay, mostly there are monocots. Where is it? Mostly monocots are there, where the endosperm is little bit left behind. Some of it is being utilized, but little bit still stays there in the mature seed as well. For example, you have coconut. So coconut which is there, it's a monocot. Okay. What about castor? Castor, even though they come under this category, but you need to understand that castor is a dicot. Okay, castor is not a monocot, it's a dicot. So this is the odd one out. That's why I said mostly. So please remember that. Cool. So I'll just repeat it. Listen to me very carefully. If at all you listen to me, this part of the story will be done, understood. So what did we have? We had endosperm. Endosperm is there for nourishment of zygote. The zygote says that I want the complete endosperm while I grow up to form an embryo. While the embryo formation takes place, it takes up all the endosperm out there. In that case, no endosperm is left behind. Okay, no endosperm is left behind. The example for these type of plants are pea, groundnut and beans. Mostly they are what? Dicots. Now on the other hand, sometimes what happens when the embryo grows up, it says that I'll have little bit of endosperm still left behind. I'll take just little bit of it. Okay, so there is persisting endosperm up there. This happens mostly in monocots, but there are exceptions. Monocots like for example wheat, barley, maize, all of it, they have something like this. But castor is a dicot, but still has what do you call little endosperm left behind. So always remember that. Guys, got it? If yes, give me a thumbs up. You guys are talking. Vijay, and that's heartbreaking. Don't talk, guys. Talk to me. I am the old person over here, papa person over here. You should be talking to me, right? Is clear? Thumbs up? Okay. Oh, now I got, yeah, yes. Cool. Now, I'll tell you one more thing. Should I be telling you now or later on? I can tell you right now. So, there are two types of seeds. Okay, there are two types of seed. Seed as in what you're talking about, ovule only. Ovule becomes the seed later on. Okay, inside the ovule, what do you have? You have the embryo. So basically, two types of seeds are there. One is albuminous and one is non-albuminous. Albuminous means you have some endosperm left back. You have some endosperm left back. And non-albuminous means endosperm is not there. Now, can you tell me the example of albuminous ones? And I just spoke about that right now. So I'm pretty sure you should be able to do it. Yes, come on, Vaishu, come on, Vijay, come on, Suvarna and Anu. Let's hear from you. What are those plants that come under albuminous, that are the ones that have albuminous seeds, where the endosperm is still left behind? And I gave you an idea about it. I just didn't use the term that's there. Come on, I'm waiting. Any idea? Ma'am, we are leaving the chat and we are going to another universe, but we won't say the answer. Is that the stand? No one knows the answer. I'll ask once more. Listen to me very carefully, okay? You know the answer. It's just that you're not able to understand it. Okay, wheat, maize and castor. The one that has endosperm. Good, Vivek. Listen, the one that has endosperm are the ones where the endosperm still persists, and we said that mostly it's monocot. Monocot means what? You have wheat, you have maize, okay, and you have what? Coconut. All of them, they come under what? This only. Another example that you have, but it is not a monocot, it's actually a dicot. That's the only odd one out, and that is castor. And that is what? Castor. These come under what? Albuminous seeds, since the endosperm is still persisting. Now the next question is, what are the non-albuminous ones? Non-albuminous ones means they do not have any endosperm left behind. Can you tell me the name of the ones which do not have any endosperm left behind? Any idea? Sorry. So Viju and Vaishu got me confused. I'm literally sorry. Oh my god, yes. Guys, I'm waiting for your answer. Over here, come on. Anu, come on. Vaishu, come on. Viju, and come on, Suvarna. Waiting for your answer. Which are the ones which have endosperm not living behind? I mean, it's not there. That's all. It's been used up completely. Here we studied, right, that completely it is used up before the seed maturation. What are those? Mostly the dicots. What are the examples for the dicots out there? Dicots means you have pea, you have groundnut, okay, and you have what? Pea, groundnut is there, and you also have beans. You also have beans. Mostly dicots come under that. Exactly, Vaishu. Pea, mustard, groundnut, all of it. Cool. So this is very, very important. Do not get confused. Later on when we talk more about seeds, this topic will come again. Okay, so I thought I'll just explain you this here itself so that you have an idea and you can relate to it. Got it? Okay, so do not forget. This is very important. If this is clear, give me a thumbs up. We'll move on to the next one. Is it clear? If it's clear, give me a thumbs up. I'll move on to the next one. Yes, clear. Give me the thumbs up. Thank you, Vaishu. Okay, now since we spoke about endosperm completely, we need to go ahead and talk about the embryo formation. Until now, we have not spoken about the embryo, right? So we're going to talk about the embryo, and while I go ahead and talk about it, you need to understand there are two kinds of embryo, dicots and monocots, right? Dicots and monocots are there. And before we go in depth about that, there's a general thing about embryo which you should know. Okay, what is it? Develops at the micropylar end of the embryo sac. What do you mean by micropylar end? So this end where you had the egg cell and you had the synergids, right? This end is known as the micropylar end. And you know the fusion happens over here. This is where you're going to get your zygote. Okay, this is where you're going to get your zygote. The zygote transforms into embryo. So we'll say the embryo formation happens in the micropylar end. This is important. So there are certain things in your NCERT which we forget, so don't forget along with that. Okay, so location, it develops at the micropylar end. Adaptation, zygote divides after endosperm formation to ensure nutrition. Once the endosperm is being formed, the zygote starts dividing. Why? It has to make sure that there is someone to take care of me, and that's why endosperm formation preceded. It formed before. Cool. Now when I say that the zygote is going to divide and it is going to form embryo, there are multiple stages of embryo, and the formation of embryo is known as embryogeny. Geny means genesis. It is going to start off. Okay, embryo formation is going to happen, and the stages of it goes like this. You would be asked question like this. You have to arrange the order in which the embryo is going to develop. So there is a first stage which is known as what, Baba? It is known as the pro-embryo. There is pro-embryo. Okay, when the one cell divides into two. Okay, I'll say now it's not a zygote anymore. It is going to be an embryo. So you have the pro-embryo. After pro-embryo, you have globular embryo. After globular, you have heart-shaped embryo. And after that, you have the mature embryo. How do you remember it? There is a easy way to remember it. Okay, there is a rap like this which you need to remember. Okay. Pro, globe, heart, grow. Okay, I know that's a really bad one, but that is how usually you might be able to remember things. Okay, you have to be silly at times. So pro, okay, pro, glow, heart, grow. Okay, pro, glow, heart, grow. So please remember, it's just that we over here, okay, there is heart, and our heart breaks, and that's when we realize that we have become mature. Usually that is the process. So you have pro-embryo, you have globular embryo, which is why it looks like round. That's why you have the name like that. It looks like a heart. That is why you have a name like heart-shaped embryo. And then finally you have the mature embryo. This type of formation is common in what? In both of your dicots as well as monocots. You have it in common in both dicots as well as monocots. So please make sure you remember the steps. If at all you do not remember the steps, you might make mistake, you might lose your marks as well. Is it clear? If this part of the story is clear, give me a thumbs up. Hello Amalu, I'm really good. How are you? Yes, guys, clear? Shall we go ahead? Clear, not clear? Yes, thank you. What about orchids? Nagaraj, they do not have the endosperm which is left behind. Okay, so they are also non-albuminous. Okay, let's go ahead then. Clear, ma'am. Fine. So now we are going to get into different type of embryo, that is dicot and monocot. Over here for your second PUC, you will be asked diagram for dicot and monocot. You need to know the diagram and I'll help you with that. Okay. And when I start explaining you this thing, there are chances that you might feel that, oh my god, I cannot do it. But trust me, it's very simple and you can do it. Know the logic and you will remember it forever. The logic goes like this. Whenever I say an embryo is developing, you need to understand that it will be having different parts, right? Because different parts is going to form different things. So the first part that you have is an embryonal axis, which looks like this. That means this is the part which is actually going to give rise to the whole plant. What do you call this as? In total, you call this as the embryonal axis. Okay, now here at the top portion you have plumule. What is plumule? Plumule is something that is going to give rise to the shoot of the plant, as in the stem, the leaves and everything out there. And the lower corner you have something else which is known as radicle. What does radicle form? Radicle helps in the formation of the root of the plant. Now since it is a root, you know that root has to get inside. Okay, it has to get inside the soil. So you need a covering for it. You need a covering for it, and that is known as what? Root cap. It is known as what? Root cap. So you have plumule over here, you have radicle over here, and then you have root cap. This forms what? The embryonal axis. Now, ma'am, why have you kept some spaces here? There's no need for having a space over here, right? There should be, a reason for it. And the reason goes like this. Listen to me. Over here at these two positions that you see, you have two things which are known as what? Cotyledons. They are known as cotyledons. Listen, here this plant is being made from the major portion over here. These two cotyledons are going to provide nourishment. Why? For the formation of plumule, for the formation of radicle, for the formation of everything. But ma'am, isn't that the role of endosperm? Endosperm should be doing this, right? What you're talking about right now. Listen, I told you that mostly in the most of the plants, when the endosperm forms, it takes up, when the embryo forms, not endosperm, when the embryo forms, it takes up all the nutrition from the endosperm, right? For what reason? For what reason? Because they want to make their own units, and their own units are known as what? Cotyledons. So these cotyledons are found with the help of what? The persisting endosperm which was there. And now since it is completely utilized, okay, it is going to completely utilize, I say that these type of embryo, okay, this type of embryo which is there inside the seed, those type of seed that contains this type of embryo are known as what? Non-albuminous. They are known as non-albuminous seeds, and mostly baba, they are going to be dicots. Dicots means they have two cotyledon out there. What are they? They have two cotyledon out there. Got it? Yes. Yes, first embryonic leaf that emerges from the sprouting seed. Yes, so we can say that cotyledons help in that. Okay, got it? Yes, and so sorry Vaishu, I think again I said your name to be something different, but yeah. So this is how it goes. Fine, so this is the embryonal axis, you have the plumule, you have the radicle, you have the root cap, you have two cotyledons. Now listen, the point of attachment of cotyledon is right over here. The top portion is known as epicotyl. The top portion that you have is known as epicotyl, and the lower portion of the attachment, this is the lower portion of the attachment, this is known as hypocotyl. So you have epicotyl which terminates with plumule, you have hypocotyl which terminates in the radicle or the root cap. Now in your NCERT there is a different picture, and that picture basically will be giving you like, oh my god, this picture does not look like the last picture, then how can we trust this? So listen, it's very easy to form the other picture. I just made it in this format so that you remember in a better manner. So the picture that goes in your NCERT looks something like this. So you have some formation. This is your one cotyledon, right, which I made like this, is basically just moving down. That's all. This is your one cotyledon that goes down, and there is your another cotyledon that moves down. And here you have the picture that is given in your NCERT. This portion which is there above, that is known as epicotyl and ends with plumule. The portion which is lower, it is hypocotyl, ends with radicle and the root cap. Now I'll show you the picture which is there so that you also remember. Listen, is the same thing, right? Is the same thing that we made. That's all. So now you know the logic and you will never forget the image also. So dicot embryo, parts are what? First part is embryonal axis with two cotyledons, right? First part is embryonal axis. The second part is basically the cotyledons out there, and you have two of them. Epicotyl is above the cotyledon level. Okay, it ends with plumule, that is the stem tip. You have hypocotyl, that is below the cotyledon level. It ends in radicle, and radicle also have what? Your root cap out there. Got it? If yes, if the dicot embryo is strictly clear to all of you, give me a thumbs up right now. What is happening over here? Come on, guys. Understanding? Not understanding? Yes, ma'am. Okay, thank you, Amlu. Only Amlu is listening, huh? What about others, guys? Come on, children. Okay, Suvarna is also listening. Thanks, thank you so much. Okay, and now we go ahead with the next one. So listen to me carefully. We have the monocot embryo now, and monocot embryo, it looks funny, right? Okay, so monocot embryo which is there, it is also super important for you to remember, but it can be a bit complicated. So again, you will need attention, and I'll need attention, so make sure you do that. Okay, hear me out very carefully, otherwise you will not understand a thing. And actually for that I need a whole different page. Where will I get a page? Okay, there's a page over here. We can utilize this. Listen, now things are going to be quite different. Again, you make the embryonal axis over here. Okay, ma'am, why did you close this? This should not be closed, right? Here you remember you had plumule, here you had radicle, okay, and the radicle is covered by root cap. That and all is there. But why did you cover this, ma'am? Because I am making a monocot. And monocot means what? Monocot means they just have one cotyledon. Mono means one. Okay, so it is monocotyledon means they have one cotyledon, and the cotyledon goes like this. They do not have two cotyledons out there. Do you have a name for this cotyledon? You just understand, whenever you do not have so many things, you just have one thing, we take care of it very nicely, right? Similarly, here also this cotyledon has been taken care very nicely. That's why we gave it a name also. Okay, it has a separate name. It is known as scutellum. What do you call it? Scutellum. And scutellum is the only cotyledon that you have in monocots. So, ma'am, you are saying that in dicots there are two cotyledons, and monocot there is only one cotyledon. In dicots they made the two cotyledons with the help of the total endosperm which was there. So in this case, since they are just making one, there should be some endosperm which should be left behind. That is the logic, right? Yes, and that is the logic. There will be some endosperm which is going to be what? Left behind. And that's why I said that mostly monocots are the ones where you will be having endosperm left behind. They will be having, that's why they are known as albuminous seeds. So the seed has this embryo, and this embryo has little endosperm left behind. That is why you call it as albuminous seeds, for the same reason. Are you getting it, children? Are you understanding it? If yes, give me a thumbs up right now. If no, ask me the doubt right now. Very, very important. Yes, perfect. Thank you, Amlu. Okay, now we are going to make the picture which is given in your NCERT, which looks something like this. So the picture that I drew and the picture which is over here, it's kind of very, very different, right? So I'll tell you how to make it simple. So the picture that I drew, it looked like this, where I had made the plumule and the radicle and all of it. Since it is a monocot, I need to understand that there could be differences. So here it's not just the plumule. Here you also have something that is known as coleoptile. What do you have? You have coleoptile which covers the plumule. P over here, P for the plumule, remember, like that. Similarly, at this particular condition, you have something else which is known as what? Coleorhiza. You have coleorhiza, R over here and R for radicle. So you have coleoptile and coleorhiza as something extra which was not there in the case of what? In the case of dicot. This is something that is different. Okay, okay. Now we will make the image that is given in your NCERT. And if you understood this, the image will also be pretty much very, very simple. Look at this. Here you have to make a veil. Okay, it does not look like a veil, but I'm telling you. This is one fin of the veil. There is the tail of the veil. And there's a small fin again over here. Okay, this is a veil. This is the veil that I make, Jasmin Mam makes. Okay, it's a funny veil. Now, Baba, listen to me very carefully. The first thing that you have at the top is what? Coleoptile. So what do? We make something like this. This is going to be the coleoptile. Cool. And I'm going to write it also so that you also understand it. Now, under the coleoptile, what do you have? You have the plumule. So let's make that. So we have the plumule right over here. What is this? This is going to be your plumule. Okay, ma'am. So then you should be having scutellum on this side. No, yes, you have scutellum. This is basically the scutellum according to your NCERT. This thing that you see is the scutellum. Okay, ma'am. Now for understanding this, what should I do? Very, very simple. Listen to me very carefully. So this part and this part, you join it up. So I'll just erase it and make it in a bit more better manner. So here you have the plumule. Okay. It goes, join up here, take it back over here, go to one tail, bring it back and join it up over here. This was your scutellum. This is your plumule, this is your coleoptile. Over here in the lower corner of the plumule, what do you have? You have the radicle. So let's make the radicle. This is your radicle. Okay, ma'am, radicle is covered by the root cap, right? Okay, make the root cap then. This is your root cap. Ma'am, then you have coleorhiza also. What about that? You make the coleorhiza. This is your coleorhiza. And that's all. And that's all. This is the picture that you need to know. Got it? As I said, if you understand the logic, picture is going to be easy. If you do not understand the logic, picture is going to be entirely difficult. Are you getting it? If yes, let me know. So coleoptile, below that you have plumule, and then you made line like this, brought it over here, joined up back. Here you have radicle, make an extra line, root cap, make an extra line, and that is known as coleorhiza. And that's done there. Got it? And over here, if at all we go back to the picture, it looks like this. There is only one cotyledon that is known as scutellum. Okay, lateral to the embryonal axis. Okay, embryonal axis is over here. Laterally, you have just one cotyledon, and that is scutellum. Radicle and root cap is enclosed with coleorhiza. Okay, epicotyl with the shoot apex and the leaf primodia is inside the coleoptile. That means that basically you do not use the term plumule, rather you can just say shoot apex, and leaf primodia is in the one that is going to form the leaf. It is basically enclosed by what? Coleoptile. Basically you are saying that plumule is enclosed by coleoptile. That is a basic thing. Cool. So do not forget it. It's going to help you out. Fine. Works. Look, this picture is very complicated, and if at all you want to remember it as such, you can. But this is again a way in which you can remember. Now one more thing is there. You see something like this. This thing, epiblast is mentioned over here. What is this? So actually the monocot also tried to form a cotyledon, but it was not successful. So we say that idea blasted off. So that unsuccessful cotyledon is what you see over here, which is known as epiblast. Okay, it is known as epiblast. Clear, ma'am. Amlu says clear, ma'am. What about others, children? It's clear? It's all cool? Will you remember it? Trust me, this is the pain point of children. They tend to mess around this particular picture, but every time monocot embryo somehow shows up in second PUC examination also. So if it's clear now, for the rest 365 days of your life, you will be in a good mood. Okay, so we go ahead. Yeah, Suvarna also says it's clear. With the next question that we have over here. So the first question that we have for today's session goes like this, and you guys can give me the answer right in the chat box. In the embryos of a typical monocot, the structure closing leaf primodia and the single cotyledon are respectively known as what? What do you call it? The structure closing the leaf primodia, okay, and the separate part is, what is the single cotyledon known as? Now over here you have got the options and you have to choose from it. Come on, A, B, C, D. What do you think is the answer for it? B. Amlu says it is B. Okay, what about others? I'm still waiting. Come on, throw your answers. It's okay. Even if you give wrong answers, I'll correct you. What more could happen? Nothing else. Okay, here in Jasmine Mam's class, there is no one to judge you guys. Okay, not even me. I'm not going to judge you. If there is someone who is very consistent with every class, I just love them. That's all. So Amalu says B and no one else is ready to give the answer. I know that you might have your personal reasons for that. But anyway, the answer for this question is going to be B itself. Why is it B, Baba? For those who do not understand and are shy to give the wrong answer, listen to me. The thing enclosing the leaf primordia we just studied, it is known as what? Coleoptile. P for plumule, P for coleoptile, remember? Yes. And the single cotyledon that you have, it is having a separate name. As I told, it is known as scutellum. That is why this is your answer for the question. Okay, so don't mess it around. And yeah, this part basically covers the root. Coleorhiza has an R for root. You can remember like that. Yes. So that's also done. And now we go ahead and talk about the seed. Until now we were talking what's happening inside the ovule. We are talking about the embryo structure. We are talking about this and that. But now we're going to talk about the ovule as a whole. It is the one that forms the seed, right? So it is going to be fun. Let's explore more about seed. The first thing is that final product of sexual reproduction is basically fertilized ovule, right? Inside the ovule you have your embryo which is being formed. So at the end, when you say that embryo is being formed, you say that fertilization has been over. And I told you, if the ovule which is there, it is fertilized, as when fertilization is over, you will not call this as an ovule anymore. You will rather call this as what? You are going to call this as a seed. So seed is nothing but a fertilized ovule. Now over here, the next thing that we have is what? Seed structure. What are the things that are there in the seed? Hello, hi Abhi and hi Shreks. Okay, good. So listen to me. What are the structures of the seed? Always understand, you had the ovule. Remember, I taught you in the previous class, you had the ovule. Ovule was covered by integuments, inner integument and outer integument. I told them that they were the sweaters that the ovule wore. Please understand, now when the ovule becomes seed, you will not call them as integuments anymore. Rather, you will have another name for it. You will call them as seed coat. You will call them as seed coat. So seed has become mature, right? And the name of these coverings which are there is known as what? Testa and tegmen. What do you call this? You call this as testa and tegmen. Testa comes from the inner integument and tegmen is basically formed from the outer integument. Okay, yes, Vaishu. Testa. Hey, not tegmen, okay, it's tegmen and testa. Yes, exactly, perfect. Okay, so let's go ahead with the next thing that is there, and that, yeah, so seed coat is one thing. The next thing that you have is what? Cotyledon and embryonal axis. Cotyledon and embryonal axis is a part of what? Of the embryo, but your seed has the embryo, right? Your seed has the embryo being formed over here, which is why we say that seed structure contains of seed coat, the parts of the embryo, that is cotyledon, and also the embryonal axis. Now, there is micropyle. Micropyle is what? I already told you. All the important things that happens is towards the micropylar end. Especially I told you that basically, Baba, your egg apparatus, that is your egg cell, your synergids are there in the micropyle. This region always remains as a pore. Why do you think it remains like that? Over here, the sweater which it was wearing, no, there was some space left behind. Why? For basically the gas exchange to take place, for the water to enter, for all of these functions. Okay, so micropyle is still there, okay, as a small pore. And why it is very important? Why micropylar pore is important? Because this is reason how oxygen as well as water is being transferred. And you need water. You need water for what? You need water for germination to happen. You know, when water is there, certain hormones in the ovule become active and they will be helping in germination, as in basically the embryo will now grow into a plant out there. So please remember the use of micropyle. This was also a very, very important question. Okay? Okay? Fine. So, Sumit ma'am from KCET is worth it, ma'am. Will I not regret take it, ma'am? You should not. Okay. You probably will not. And so you're not excited for me, Sumit? Dun, dun, dun. Okay, it's okay. So let's meet. Even I am excited. I'm excited to see your excitement. Okay, anyways. So guys, whoever is talking amongst yourself, please try to include this papa friend of yours. That is me also. Otherwise, I'll get bored. You imagine my plight. I am just teaching you guys, and there is no one to talk to me. There is only one camera, and you guys are talking amongst each other. How will I feel? I'll feel very bad. So please talk to me and listen to me. Okay. Okay. Fine. So until this, I hope it's clear. Second part of the story, we spoke about types of seeds. We already spoke about it. What is it? Non-albuminous and albuminous seed. Non-albuminous means what? Where the endosperm is completely utilized. How is the endosperm utilized? To form the cotyledons out there. Example for them, we discussed about it already. It is what? Pea, groundnut, mostly all the dicots out there. Then you have one more one. That is what? Albuminous, where the endosperm is still there. Right? Endosperm is still persisting. This mostly happens in the case of monocots. Example is what? Wheat, maize, barley, etc. But there is a dicot which comes as an example. That is the only dicot which is given in your NCERT as an example, and that is castor. And that is castor. Cool. The next thing that we have over here is what? Perisperm. What do you mean by that? Children, are you understanding this thing? Or is it me who is just talking? We're talking about seed, okay, and in seed we said that there is micropyle still left. Why? There's a small pore for easy exchange of gases as well as water for germination. We spoke about the type of seeds, albuminous and non-albuminous initially itself. Now I'm going to talk about one more type of seed, and this is very important, super important, super into 100x important. And what is that type of seed? Listen to me carefully. So usually what happens, when I say there is ovary and there is ovule, there are certain cells over here in the ovule. These cells are known as cells of nucellus. Okay, and one cell among them actually gave rise and basically formed the embryo sac, and now you have an embryo out there. Usually what happens is that the cells of the nucellus is being used up while all this process happens. It does not sustain until this particular time. Okay, but sometimes the cells of the nucellus, they persist. They persist for what? They persist and say that one day embryo will be formed and we will also nourish them. Okay, we are gonna also nourish them. You alone won't be able to do that. I will also nourish them. Well, when this layer is there inside the seed, you call this layer as perisperm. You call this as what? Perisperm. And the example for seeds which have perisperm are two of them, and it is super important. What are these? The two important ones are what? B square. B square as in beet. One is beet that you have, and the other one, anyone can guess? The other one which you have, anyone can guess? Beet is there, and there is black pepper. And there is black pepper. So mostly the cells of the nucleus are not there, but sometimes they persist. If they persist, you call this as perisperm. Okay, this layer is perisperm, and this perisperm which is there, it is found in two of them according to your NCERT. It's B square. One is beet and the other one is black pepper. Exactly, Amlu. It is what? Black pepper. Amlu said, guys, please stop talking and listen. Exactly, that's what I want to talk. Che, that's what I also want to say. Guys, please do not talk to each other. Yes, black pepper, exactly, Vaishu. That's my children, that's my machas out there. So that is how it goes. Okay, now listen to me. There is one more term which can confuse you guys, and that is pericarp. Pericarp. Anyone has any idea what do you mean by pericarp? You know perisperm, I just spoke about it. Do you have any idea about pericarp? Have you heard about it before? Any idea? If you have not, say no, ma'am. If you have yes, give me the answer. It's okay if at all you say no, because that's my duty anyway. I have to teach you this particular thing out there. Perisperm, we know. Pericarp is something that we should know, because in your second PUC examination, they ask you the difference between pericarp and perisperm. And Papa children, they are like, oh my God, I do not know. It's all so confusing. This chapter is boring as well. Why will I remember all of it? How will I remember all of it? So I think it's a no, because there is no answer coming up. So listen to me. This, I'm talking about ovule, in the ovule, I'm talking about what do you call the layer out there. But when I talk about pericarp, I am basically talking about ovary. I'm talking about ovary and not the ovule. Ovule becomes a seed. What happens to the ovary? Ovary becomes what? It becomes the fruit out there. The ripened ovary wall of a flower that forms the outer layers of fruit. Exactly. And Amalu also said epicarp, mesocarp, endocarp. Oh my God. All of you know, Vaishu and you, very good. Now listen to me. We're talking about the fruit that is ovary, and there comes the role of pericarp. So Baba, all of us, we know that now it's mango season, right? And we all might have eaten mango also. The outer portion of the mango that you just peel off, okay, that you just peel off, it is known as what? Epicarp. It is known as epicarp of exocarp. Okay, epicarp or exocarp. Now Baba, you have one hard thing over here, right? A very hard thing over here. What do you call this as? This is your endocarp. What do you call this? This is your endocarp. Inside the endocarp, what do you have? Inside the endocarp, you will be having your ovule. That is nothing but the seed. Inside the seed, what you will be having? You have the embryo, all of it. So seed has its own covering, that is testa and tegmen, that is all there. But this hard shell that you have, okay, it is a part of the fruit only, that is a part of the ovary itself, and that is known as what? Endocarp. And the middle portion that you eat, yum, 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 what do you call that as? That particular thing is known as mesocarp. It's known as mesocarp. So epicarp, mesocarp, endocarp, all put together, they are the walls of the ovary, which you actually call as pericarp. Which you call as what? Pericarp. So what is perisperm? Perisperm is nothing but the remnant, okay, the remaining cells of nucleus which is there for providing nourishment to the embryo. That is perisperm, found in black pepper and beet. Pericarp is what? It is basically the outer covering of the ovary walls. Over here, it is known as pericarp, and basically you have distinction amongst them. Okay, there is epicarp, there is outermost layer, mesocarp, there is a fleshy layer, and endocarp, that is the hardest layer which is there. Cool? Got it? Amlu and Vaishu, everyone? Clear? Understanding? If yes, give me a thumbs up. Ayyo, where is the live? Perfect. Cool. Okay, so we were talking about seed, and slowly we transitioned into what? We transitioned into fruit. But there is one more thing about seed that you need to know. When seeds mature, its water content is reduced and seed become relatively dry. Okay, so whatever water content is there, it becomes reduced and it becomes very, very dry, relatively very dry, almost 10 to 15% moisture by mass. 10 to 15 moisture by mass is the thing that is left behind. The general metabolic activity of the embryo slows down. So whenever you say that a seed is there, you need to understand that a seed, if at all it is not getting all the good conditions, okay, if it is not getting enough amount of water, this and that, basically it cannot grow, which is why it will be metabolically less active. It says that I will not work anything, I'll do the work later on. So please know that. Okay, so the things that you need to remember about seed were, seed coat is there, testa and tegmen. Other part of seed is what? Basically, you're talking about cotyledons and the embryonal axis. You said that the seed will be having a pore, that is for oxygen exchange and also for water transport during germination. Then you spoke about the type of seeds. There are two, non-albuminous, albuminous. Non-albuminous means no, it's not there. What is not there? Endosperm is not left behind. Everything is being used up. Albuminous means they have endosperm left behind. Non-albuminous, under that comes basically mostly dicot plant that you can break into two. So the pea, everything that you break into two is basically cotyledons, nothing else. Okay, so pea, groundnut, beans, all of them non-albuminous. In albuminous comes usually monocots. For example, wheat, you cannot break into two. Maize, barley, you cannot break into two. Castor is a dicot actually, but it comes under albuminous. So remember that. Cool. Then we studied that basically sometimes some part of the nucleus remains back. You call it as perisperm. That is there for providing nourishment, and the example for that is beet and black pepper out there. Okay, cool. And then we spoke about pericarp, and yes. So now you have a question. Persistent nucleus is found in what? Persistent nucleus is found in what? Black pepper, beet, mustard, or both A and B? What do you think is your answer for this particular question? What do you think is the answer for this particular question? D. Amalu says D is the answer. What about others? Vaishu, come on. And others, if at all you're watching this video, is D the answer for this question? Well, the answer for this question is D. It's both A and B, that is black pepper as well as beet. So you guys are absolutely correct. Cool. Okay, Suvarna also said D and Vaishu also said D. My dear little children, you are too good. And I'm super proud of you. Good. Perfect. Let's go ahead then with the next thing that we have, and that is advantages of seed. Before we go ahead with fruit, there's a little more about seed that we need to remember. And yes. So what are the advantages of a seed? Again, your second PUC question which usually comes in. First is independence from water. Pollination and fertilization do not rely on water. Listen to me very carefully. Initially, you have bryophytes, pteridophytes and everything. They needed water to be they needed water for fertilization and everything to happen. But when you talk about angiosperms, flowering plants, they have seed. And seed formation does not require water. So there is independence from water. That means that they can be grown anywhere very easily. Okay, it does not need water. So pollination and fertilization do not rely on water. That is the first and the best part. This is a good thing about gymnosperms because gymnosperms have seed. Second thing, dispersal and colonization. You understand, seeds are very easy to be dispersed. For example, I have some seed in my stuck somewhere out there and I move around. Basically, it's very easy for the seeds to be dispersed. If at all the seeds were big, this large, can I transport it? No. Even unknowingly, I cannot do it. So you get it? So that is the thing. Even when plants or organisms comes to eat certain things, on their surface, some seeds might stuck. And basically, when they move around, it can be easily dispersed. So dispersal and colonization is the next thing that we have. Now, the very next thing that is there, that is, if at all you want a plant to be there, you need the embryo to be safe, right? And you need the embryo to be fed with food, right? So seed has the embryo, and seed, what does it do? It makes sure that the embryo gets all the food. So it can be in the form of cotyledons. Okay, they have enough food reserves. The seed makes sure it has enough food reserve either in the form of cotyledons, or basically it can be in the form of endosperm. The endosperm itself is the one that helps in the formation of cotyledons, right? So either it can remain as endosperm as in monocots, or it can form multiple cotyledons as in the case of dicots out there. But anyway, they are very helpful. The last way, again, seed has seed coat and everything, which provides what? Protection. It provides protection. That is the fourth thing. So independence from water, dispersal and colonization, food reserve, and basically they provide protection. And the last but not least, they help in increasing the genetic variation, because seed is formed when male and the female gamete comes together, and mostly they are from different plants, mostly. So obviously when they come together, it can give rise to different, it's not of the same plant, mostly of the different plant, so it can give rise to more genetic variations as well. So that is an additional point. If at all we want to write, we can write it. Cool. This part of the story clear? Advantages of seed, is it clear? It's not like a question that comes with 5 marks and all, but sometimes a 5-marker question can be divided into 2 parts, 3 plus 2. It can be 2-marker. So you should know about this. And these are things that we feel that, what is it? We'll just ignore it. So don't do that. Okay. Cool? Yes. So Amlu gave me thumbs up. So I believe that I can go with the next part of the story. So for the next part, I'm really, really excited. So over here, I have a picture. This picture, this is a plant. Okay. And over here, there is another plant that is this. And I'm pretty sure at least you can guess this. Can anyone guess which is this particular plant? Thank you, Suvarna, for giving the thumbs up, Baba. Which is this particular plant? Any guesses? It's a very famous plant. We eat its content out there. Anyone? It's very sweet when we eat it. Any idea? No idea? Very sweet. We get it in the dry fruit packet also. I think it's loading. Slowly you might be telling the answer for it. No one? Okay, then I'll only say just a second. So it's date palm. Okay, it's what? Date palm. And you might have had dates before. And date palm is very, very, very important over here. Why? Because Phoenix dactifera or basically date palm has seeds which can remain for a very long time. The seeds can be remaining as in viable, as in they can, suppose over here as of now they have not getting enough amount of nutrition or nourishment or water to germinate and things like that. They can stay like that and later on they can germinate. And can you imagine, they are the ones that germinated after 2000 years of gap. I mean, they can remain viable for almost 2000 years, and that's crazy. Please remember it. It is what Phoenix dactylifera, or also you call this as date palm. They were discovered in Dead Sea during the Dead Sea excavation near King Herod's place. Okay, King Herod's place, and as I said, during the Dead Sea excavation that took place. Now, similarly, you have Lupinus articus. This is another type of plant. And here the seed of this particular plant stayed viable for 10,000 years. It is what Lupinus articus. They are usually found towards the Arctic tundra where it is very much cold. Okay, so you have to remember these two names. These are very important in terms of KCET questions as well. Okay, so first topic is seed longevity, as in basically they're talking about for how much time the seed can remain viable, for how much time can it be still okay. It's still okay that after some time, if at all I give water, it can grow up. That is what you mean by viability. So, or germinate. So some seeds lose viability in months while others last for years. And the examples for the oldest viable seeds are this. First one is Lupinus articus. It remains viable for 10,000 years. The second one is what Phoenix dactylifera or date palm, which remained viable for 2000 years. So please remember this. This is very, very, very important. You can get questions on the basis of it. I have shown you the picture so that you can remember better. Okay, so date palm, just remember that King Herod was eating date palm, and it was 2000 years old viable date palm seed. Cool? Clear? Is this particular part of the story clear to all of you? If yes, let me know. Just a second, children. Okay. Clear, ma'am. Okay, thank you so much, Suvarna and Amlu and everyone. Okay, cool. And now we go into what? Fruit, and also we'll talk about false fruit. Listen to me carefully. There are two types of fruits. True fruit and there is false fruit. What do you mean by that? So generally I told you about this very important thing, that is what? We get fruits how? When the ovary is there and the ovule is there, the ovary becomes the fruit. That is the basic logic, and the ovule which is there, it becomes the seed. This type of fruits where the ovary itself, okay, or ovary is the only one which is participating in the fruit formation, they are known as what? True fruits. They are known as true fruits. On the other hand, suppose, okay, just understand that this is the female part that you have, and this female part of the plant rests on the thalamus. Here you have petal and everything, and this is what? The thalamus. Suppose the ovary which is there, it takes part in the formation of fruit, but along with that, the thalamus also helps in the formation of fruit. So it's not just ovary alone which is going to form the fruit, it's also some other parts. This type of fruits are known as what? They are known as false fruits. What do you call them? They are known as false fruit. So you have true fruit and you have false fruit. True fruit is where the ovary which is there, it's the only one which is participating in the formation of fruit. False fruit is where the other parts are also participating in the formation of the fruit. Now, ma'am, I want example for this. Apple is an example for this. Apple is the best example for the false fruit. So the thing that you actually eat in the apple, okay, the thing that you actually eat in the apple is basically the thalamus. So, ma'am, ovary does not take part. Ovary also takes part. So, the parts of the ovary are basically within inside. You might have seen that it's very tough within, right? So, that is the part of the ovary which is there. The rest of the part that we can easily eat is actually the thalamus, which is why we say that basically apple which is there, it's a false fruit. Apart from that, you have cashew. Apart from that, you have strawberry. These three things which are there are false fruits. So please remember, you have apple, you have cashew, and we have strawberry. So ACS, apple, cashew, strawberry, they are all false fruits. Now it can be thalamus, it can be other parts of the plant also which can help in the formation. Cashew, strawberry, apple, false fruit. Exactly. And Suvarna, yes dear. Amalu, perfect. Great. Okay. So over here, I'll just read about it so that you get a better understanding of it. So where it does this start? So just wait for a second, we'll talk about this later. First this. True fruits developed only from the ovary, most plants. False fruits, thalamus contributes to the fruit formation. Example, so it's not just thalamus, other fruit parts, other plant parts, okay? Don't just write thalamus, it can be other plant parts. In the case of apple, it is thalamus. In some case, it is pedicel. In some case, it is pedicel. Pedicel means, you know what? Over here, you know, your stalk-like structure is there. Then you have thalamus. Then you have the flower. Pedicel also sometimes contributes. So it's not just thalamus. It can be any other plant parts apart from ovary, which helps in the contribution of it. Cool? That is false and true fruits. And I hope you will remember the example: apple, cashew, strawberry. Now, at the top they have mentioned about one more thing, that ovary becomes a fruit, that is okay. Some fruits are fleshy and some fruits are dry. What do you mean by that? In the fleshy fruits, you will be finding what? The mesocarp to be there. In the case of some dry fruits, for example, when you talk about groundnut and mustard, they do not have a fleshy kind of ovary. It's very dry and you can just peel it off. You get it? So yes, they have their own ovary which transforms into a fruit, but over here it's not fleshy, rather it's dry. Please understand that. So fleshy as in the case of mango, in the case of guava, but dry in the case of what? Mustard, in the case of groundnut. Cool, so that is also there. Ma'am, when can we expect CET results, cosmic? Probably by this particular week, or it could be going to the next week, but most probably it should be coming by the end of this week, is what I believe. The reason being that, you know, what do you say, on 1st, that is today, the examination for the agriculture practical should be over, and they will need some time to analyze the results of that as well, and post that they should be giving the results. So it should not take much of your time. Yeah? Yes. Cool. And now we go ahead. I hope this part of the story is completely clear, and we go ahead with what? Parthenocarpic fruit. Okay, thank you, ma'am. You're the nicest. Oh my God, that's so sweet of you. Thank you so much. Okay, now listen to me. There is one more very important term, a very big term which is there, and that is parthenocarpy. It's very important, such that we should not be losing out on this particular thing. It's important and we'll learn it. Cool. Before that, this is a picture of apple that is given in your NCERT, and they have clearly mentioned the thing that we eat is actually the thalamus, and over here you have the seed, and just outside the seed you see endocarp, mesocarp and everything. This is a part of the ovary. So ovary is also there, but inside. Please know the labelings. These are very, very important for you guys to remember. Okay, now we come to parthenocarpic fruit, and when we talk about parthenocarpic fruit, or when we talk about apomixis and everything, there is something that you need, and that is attention. Attention to details, otherwise we might mess around. So listen, I'll explain both of them together over here. Let's start with parthenocarpy first. Or for a safer zone, just a second, I'll just remove this picture. Okay, so over here I have parthenocarpy, and this is going to be Apomixis. Cool? So parthenocarpy, it goes like this. You have the ovary, you have the ovule. Now what have we studied? We have studied that after fertilization, after the male and the female gamete fuses, the ovule transforms into the seed and the ovary transforms into the fruit. What if I tell you What if I tell you there is no fertilization that is happening up over here? That is the first condition that you need to know. No fertilization means, do you think seed will be formed? No seed is going to be formed. Okay, that means no fruit is going to be formed? No, that's not there. Fruit, ovary will still form the fruit. Ovary will still form the fruit. How, ma'am? How is that even possible? Because there are certain hormones which are going to play a major role over here. It's going to ripen the ovary to form the fruit. It's because of hormones. Okay, you need to understand that. And now, since there is no fertilization over here, do you expect any seed to be formed? No. So ovary will form the fruit, but this fruit, Baba, will not have seed. It will not have seed. How can that happen? As I said, with the help of hormones. Now listen to me. Parthenocarpy which is there, can we induce it? Can we make plant undergo this particular thing? And the answer would be yes. It would be definitely a big yes. The reason being that is what, my dear children? How do you say that the plants can be induced with parthenocarpy? How can you do that? By giving some hormones. So there are parthenocarpic fruits. You might have seen in plants there are certain fruits which are being formed without any seed. For example, grape and all. How? Basically, there are some hormones which are being put out there. Like that only they can undergo parthenocarpy, or basically they will be forming fruit without the seeds. And the best example of naturally it happening is what it's seen in the case of banana. Banana, have you seen seeds? It's very remnant, small thing that is there. You get it? So this is very, very, very important. Cool? Works? So children, have you understood parthenocarpy now? If yes, give me a thumbs up. If you have understood it, or if you have any doubts, please ask me right now. I will be helping you for sure. 10,000% sure. Yes, Amalu has understood it. Suvarna also has understood. Thank you so much. Cosmic, ma'am, is there a way I can reach out to you for info? There's only one way, and that is YouTube, dear. Okay, let's go ahead and let's talk. I'll try my best to reply to you, dear. It's not like I will not. So please ask me the comments, sorry, your doubts in the comment section. I'll try my best to reply to it. Okay? Yes. Now comes apomixis. So here, what is happening? Parthenocarpy, in short, I can say fruit formation without fertilization. Fruit is being formed without fertilization. What is apomixis? Apomixis is going to be a bit different. Okay, in apomixis, what is going to happen? Let's see, and you have to pay your attention over here. You have the ovary and you have the ovule. You have the ovary and you have the ovule. Fertilization should happen, right? Fertilization should happen for this ovule to become seed, but here things are going to be different. You do not need fertilization for seed to form. So I'll say that basically over here, without fertilization, seed is being formed. Here, without fertilization, what was happening? Fruit was being formed. How can that be even possible, ma'am? That might be your question. So listen to me. Inside the ovule, I said there is embryo sac. Inside the embryo sac, you have what? The female gamete. Female gamete is usually haploid in nature. Then the male gamete comes, that is also haploid. Haploid becomes diploid. But in some condition, what happens is that there is not a haploid egg over here, rather it's already diploid. The cell over here is already diploid. Now if the female gamete, this is one way, okay, there are multiple other ways that ovule can become seed. This is one way. Female gamete is already diploid in nature, and if it is diploid in nature, do you think it will need a help of a male gamete to form zygote? It says that, Macha, I do not need your help. I do not need your help. It is already 2n in nature, and that's exactly we wanted. This thing can later form embryo by itself. The cells can divide and it can form embryo by itself. Do you think fertilization happened over here? No. Will the ovule turn into a seed? Yes. And if the ovule is turning into a seed, ovary will also turn into a fruit. But basically, this is the logic out there. Okay, there is no fertilization needed, but seed formation definitely happens over there. This is very, very important. Now, when I say that something like this is happening, you should understand one more very important thing. Is there a male gamete involved? No. And when only one parent is responsible for the formation of embryo, can I just say it's just like asexual reproduction? Because only in sexual reproduction you need gametes. But if there is no gametic fusion over here, I'll say apomixis is like asexual reproduction itself. Yes, can I say that? I can say that. So I'll say it's just like asexual reproduction, and basically whatever features are over there, okay, whatever features are over there in this particular plant, the same features are going to see in what? The progeny. Progeny will be similar to only one parent. The progeny is going to be similar to one parent out there. I hope this is clear. Yes? Got it? So Amalu, yeah, Vaishu is saying apomixis is a type of asexual reproduction in plants where seeds develop without fertilization. Exactly, exactly. Okay, and Amalu is saying it mimics sexual reproduction. Yes, how? Because it is doing what? Embryo formation and things like that. Cool. Now one more very important thing. That's not all. Okay, this is not all. This is not the only way you can do apomixis. There is also one more way. And what is the one more way? Remember I told you sometimes in ovule the nucellus is persist. The cells of the nucellus, they sometimes persist out there. This is the second scenario. What happens is that the cells of the nucellus are normal cells. They are again diploid cells, Baba. They are what? Diploid cells out there. Sometimes the diploid cells which are there, they get inside the embryo sac and they divide. And when they divide, they are actually going to form what? Embryo out there. They are going to form embryo out there, and mostly they form multiple embryos. So we can say cells of nucellus gets inside the embryo sac. I know it's complicated, but it happens, and form embryos. And when they do that, you need to understand one more term comes into the picture, and that is known as polyembryony. Polyembryony. What do you mean by that? Multiple embryos are being formed. That is why you use the term polyembryony. I'll not say that this is the way that apomixis happens. No. Apomixis have two ways in your, according to your NCERT. One is the female gamete is already diploid. It can just divide and form the embryo. Second is basically the cells of the nucellus get inside and they form multiple embryos. When this happens, you call this condition as polyembryony, and this is, okay, this is very important. This is mostly, just a second. Yeah, so yeah, this is just something which is happening otherwise. Okay, so please remember that. Yeah, sorry. So polyembryony which is there, it is mostly seen in the case of what? In the case of citrus fruits. In the case of citrus fruits. What are citrus fruits? For example, I talk about orange, I talk about something else. They are the citrus fruits out there. Okay, orange, mango, they all come under citrus fruits, and polyembryony is seen up there. Cool. Is it clear? We'll learn more about them. It's not like I'm ending up over here, but I hope you now understand the difference between parthenocarpy and apomixis, because this is one very important thing that children get confused in also. Clear? Okay, thank you so much. And now we go ahead and we talk more about it. So where are we? Yes, parthenocarpy is what? Develop fruit, okay, develop without fertilization. Parthenocarpic fruits are what? They develop without fertilization. Example is banana. Now it can be induced also. It can be induced using growth hormones, and in that way you get what? Seedless fruits. You are going to get what? Seedless fruits. Done. Now we talk about apomixis, which is there. Apomixis which is there, and you need to pay attention. There is more to it. First one, apomixis is seed formation without fertilization. Where do you usually see it? Asteraceae and grasses. Asteraceae and grasses, this is very important. Asteraceae and grasses is where you usually see apomixis. Now, it mimics sexual reproduction, but it is actually a form of asexual reproduction. What is it? It is mimicking sexual reproduction. Why? Because there is formation of embryo and stuff like that. But it is actually a form of asexual reproduction because only one parent is involved. Now, there are two ways. One is the egg cell is already diploid. The diploid egg cell form without reductional division. They develop into an embryo without fertilization. So basically, usually egg is being formed, egg cell is being formed after reduction division. But over here, diploid egg cell is being formed without any reductional division, right? And this diploid egg cell later forms the embryo without fertilization also. This is one way. Second way is nuclear cells. Nuclear cells which are there, they divide, they enter the embryo sac and they develop into multiple embryos. This is found in the case of citrus fruits such as mango, orange and things like that. And this condition is known as what? Polyembryony, where more than one embryo is being formed inside the, inside the embryo sac. Cool. Yes, what is the genetic nature of the apomictis seeds? They are going to be exactly like the parent plant, clones. They are going to be exactly like the parent plant which is there. Now, difference between parthenocarpy and apomixis results in production of a seed, whereas parthenocarpy results in the production of seedless fruit. Yes, that is there, but you have to add up. Okay, production of seed without fertilization. Without fertilization term is super important for both of them. But good version. Okay, so this is important. Please add without fertilization. Now, in your NCERT, one more thing is given, and that is important. By the way, this is how multiple embryo formation would look like. Okay, anyways, one more important thing is there, that how are farmers able to utilize it in the best way? This apomictic seeds, is it good, is it bad, things like that. So listen to me carefully. Suppose I am the apomictic seed. I am the apomictic seed, I am the mother, and I gave rise to the child out there, just like me. Suppose I had all the good characters, I am pest resistant, I am drought resistant, I am this, I am that, whatever is there. Now what happens is that my child is going to be just like me, the new plant which is there is just like me. So isn't it good? Yes, it is going to be really, really good in that particular case. On the other hand, if at all I relied on sexual reproduction, what is going to happen? The male will also contribute to something. It could be bad, right? It could be bad. So this is not applicable for all the plants, but you need to understand if there are some plants which are really good, there's superior variety of crops. If at all, we make them produce apomictic seeds out there, and basically we do not have to be worried. These seeds are going to produce within themselves itself. All the good character are going to be just going on. So that is one very important thing that is there. Second thing, if at all we talk about hybrids, when I say this plant and this plant is crossed, in the generation which is there, and when in the future they are going to again undergo sexual reproduction, there are chances that the good character can just go off. So that again is being very much reduced. So importance in the hybrid crops is that hybrid loses traits if grown from the normal seeds. Okay, hybrids which are there, if at all they are growing from the normal seeds, it is going to lose up all the traits. Why? Because normal seeds undergo sexual reproduction where two parents are involved. As I said, over here in the case of apomictic seeds, since only one parent is involved, and if that parent is really good with good features, it's all cool. I do not need to be worried about anything. Second, apomixis prevents segregation, allowing farmers to reuse hybrid seeds. As in sexual reproduction, there are chances, as I said, it can be mixed up, but here it's not. The second reason, rather, I'll say both of them seems like the same point itself, so I'll add up on a point and I'll tell you. Hybrid seed production is very costly. Right, you have to take the plant, you have to cross it, and basically need to have multiple plants like that to produce something like that. It is very costly. But if at all you have apomictis seeds, it's not costly. Once you have a plant which produces apomictis seeds, it just keeps on going. So it is cost efficient as well. So there are 2 points. First point is that over here, in the case of apomictic seeds, there is no segregation that is happening. It will be just like the parent plant. If the parent plant has the good features, it is just going to go on. Second is, it is cost effective. Why? You do not need to buy multiple hybrid seeds or hybrid plants again and again. It's all cool because once you have got it, basically they are going to reproduce within themselves itself. Got it? So these 2 points are something that you can easily write up about the uses of apomixis. Clear? Hiya. Clear? Yes, perfect. Now, I think I have a question. Yes. What is common between vegetative reproduction and apomixis? What is common between vegetative reproduction and apomixis? So over here you have got options A, B, C, D. A, both are applicable to only dicot plant. B, both bypass the flowering phase. C, both occur around the year. D, both produce progeny identical to the parent. What do you think is the answer for this? Vegetative reproduction means what? You're talking about asexual reproduction, vegetative parts. Now the reproductive part, vegetative parts. For example, you might have seen bryophyllum. In that, the small from the nodes you get small from the notches you get small parts, and they fall up and they give rise to the whole new plant. It's the leaf which is producing it, right? It is vegetative part, not the reproductive part. So over here, vegetative reproduction and apomixis, if at all you have been asked what's common between both of them, what would be your answer? Come on. B. Amalu says B. Okay, what about Suvarna and what about Vaishu? Any idea? Okay, Suvarna also says it is B. Let's see. So both are applicable to only dicot plants. Wrong. Both bypass the flowering phase. Okay. Both occur around the year. Both produce progeny identical to the parent. Are you sure it is B? Are you sure it is B? Or it could be D? Flowering phase can happen, no? Flowering phase can happen in vegetative reproduction also. I mean, the plants can be vegetative, can produce flowers, but it can choose not to produce through flowers. But it can happen. There's no, I'm not saying that in vegetative reproduction there's no plant that has no flowers. It can have flowers, but it chooses to have asexual mode of reproduction, right? This would be the answer, right? D would be answer. Both produce progeny identical to the parent. Why? Because both are asexual mode of reproduction. They are just going to be looking like the parent plant. Did you get it? Did you understand the logic, or do you have confusion? If you have, you should be asking me right away. That's not at all a problem. Got it? Yes. Okay, so D is the answer for the question. Not get it wrong. This could be important for you. Now, one more question was there. Find the mismatch. Find the mismatch. And this will be the last question. Explain once more. Yes, perfect. I can explain once more. That's what I like. Ask me questions. Okay, so you have been asked about what is common between vegetative reproduction and apomixis. In vegetative reproduction, it's asexual mode of reproduction that is going to happen. That means vegetative part. For example, I have a plant over here. This particular plant has leaves and it has flower. Here the flower might be doing asexual reproduction, but the leaf is also contributing and doing, it might be doing sexual reproduction, but here the leaf might be doing asexual reproduction. This is known as vegetative propagation, right? Yes. Apomixis is also asexual because only one parent is involved. So if at all I say both are applicable to only dicot, that will be wrong because that's no necessity. It can be monocot or dicot. Both bypass the flowering phase. Here it can happen. Flowering phase could be there, but you're depending on the other parts. Okay, so there is no necessity that flowering phase would not be there as such. You need to understand that. Again, over here in apomixis also, Baba, you have the seed, that is the ovule and the ovary. Where is it? It is inside the fruit only, no? Sorry, fruit nahi, flower, no? The flower is there. Okay, the flower is there. Inside this only you have all the parts of the plant, stigma, style, ovary. Inside the ovary you have the ovule where all of it is happening. So flower can be formed there. So even if you think that in vegetative reproduction you're not talking about this, ma'am, you're talking about this only, then fine. But when you talk about apomixis, there you have a flower where all of this thing is happening all together. Right? So which is why I'll say this would be wrong statement to say. Now both occur around the year. We do not know. Okay, and it may not. Both produce progeny identical to the parent. This is pretty sure. This is a pretty sure thing. Over here, this is going to produce exactly similar progeny over here because it's a vegetative part. There is no sexual reproduction. It is asexual. Only one parent is involved. Definitely, it is going to look like the progeny. Okay, that means the progeny is going to look like the parent plant. Similarly, in apomixis, only one parent is involved, which is why the progeny will again look like the parent itself. Got it? Now, or do you need explanation? I can still explain. Perfect. Got it. Thank you. Fine. Now question, find the mismatch. And it goes like this. A, false fruit other than ovary thalamus taking part in the fruit formation. B, parthenocarpic fruits developed without fertilizers. C, true fruits, fruit developed from the ovary. Seeds developed from the ovary. What do you think is the answer for this? You have to find what? You have to find the mismatch. You have to find the mismatch. B is the answer. Amlu says B is the answer. Okay, what about others? What about others? B. Suvarna also says B. Exactly. So false fruit are the fruits where other than ovary, other parts of the flower are also taking part in the fruit formation. That is actually correct. Parthenocarpic fruits are the ones where fruits are being formed without fertilization, not without fertilizers. Fertilizers, manures, they provide nutrients, right? So this would be a wrong statement. Okay. So now true fruits are the fruits which developed from the ovary, and seeds are what? They developed from the ovary. That is seeds developed from wait. Seed should be developing from the ovule, right? So this will also be a mismatch. Wrong question, wrong question, wrong question. Alert. So both this statement would be wrong. So I think there is a typo error over here. It should be ovule. Okay, while typing it could have been different, but yes. But you guys didn't note it, only I did. Okay, so yes. Anyways, so B is the answer for this question, if at all that is ovule. Cool? Got it? Here also I'll write it. Yeah, it's written. So that's about it. Were you able to understand all of these things in this particular class? Do you think it was helpful for you? Or do you think no, it was not that great? Well, I'm pretty sure you will not say that on my face, but but my dear children, I just want to tell you one thing, that is I adore you guys, and I am really proud of those of you who attended. It might be a very less number, but I do get it that no one would be pretty serious at this particular point, but you are, and you attended this class, and I am super proud of you for that. Make sure you complete all your things which is there around it. Okay, it's very, very important. As in, what are the chapters that the chapter that I'm doing right now, if at all you have heard this, I think that you might be able to understand the other parts that I have done. Complete those. It will be really, really good for you guys. Cool? Very helpful. Thank you so much, Amalu. Okay, so that's all for today. And also there is Pariksha Veera batch which is going to start by June mid, most probably, and if at all you're interested, you can check out the link that is given in the description section of this particular video. Yes, Yalla Miri, yes, tell me dear, Akka, I have a doubt. And I hope all of you have hit on the subscribe button. Please do that. It could be really helpful. Yeah? If you think that it was useful. So children, can I just go? I hope that works. And do not forget to hit the hit the like button if at all you think that this helped you. Okay, so I think it's time to leave. So bye-bye. Thank you, Suvarna, and thank you, Amalu, and thank you, Veshu. Means a lot. Lots of love from Jasmin Ma'am and Yellam. Yuri, if at all you have any doubt, put it down in the comment section. Okay? Bye-bye.